Caprinae subfamily is a convoluted mess to sort out, consisting of 2 species of Pseudios, 6 sorrel, 4 goral, 1 mountain goat, 7 ovis, 8 ibex, 2 Caucasian tur, 1 Barbary sheep, 3 tahar, and 1 markhor. Ancient biblical wording from Hebrew indicate that sheep and goat belong to a basic type or group belonging to the same kind in monobaromology. I was still able to determine the original created kind even through this convoluted mess. I have determined it is the wild Benzora ibex, or the Persian ibex. This was the original goat of today. Let's begin with recent studies based on mitochondrial DNA, which prove the West Caucasian tur appeared to be more closely related to this wild goat than even its own Eastern Caucasian tur sister. This new genetic evidence conclusively proves that the Persian ibex wild goat is related to the genus, which until recently was not even considered related. The markhor as well is related. It is only relatively separated by a little bit from other goat forms. Previously it had been considered a separate branch of the genus altogether. The Perineum chamois, which descended from the wild goat, gave rise to the chamois, which speciated into the growls and sorrels of today. New mitochondrial chromosome B testing and nucleotide substitution rate testing prove this. So much for the growls and sorrels coming first, like they assumed and told us for decades based only on fossils. And since we have validated genetically that the alpine ibex descended from the Persian ibex, then it is clear and obvious that we have narrowed down the basal ancestor for all living sheep and goats to the Persian ibex. Specifically regarding sheep, the wild goat, known as the Persian ibex, gave rise to the wild mouflon, which speciated into all the modern-day sheep we see today. Based on comparison of mitochondrial cytochrome B gene sequences, all 6 subspecies of sheep have been identified now to the Siberian snow sheep, bighorn or Dall sheep of North America, agrali of Central Asia, and the domestic sheep of Eurasia. Urial is found in the western Central Asia and northern eastern Iran and western Kazakhstan to Pakistan, all the way to India, and traced back to the mouflon. So we can be sure now as well that the wild mouflon gave rise to the modern-day sheep, because genetics has proven this, which again should be the primary indicator of relation before one resorts to presumption via the fossil record. All modern domestic day sheep breeds descended from the ancestor of the Parisian ibex, the mouflon, and all domestic sheep today are from the mouflon and are numbering over 1 billion worldwide. You see, it is taught by evolution that multiple waves of independent migrations of wild goat had to have taken place in Europe. This is because of their assumption of old-scale timelines must be true, and this is why they rely so heavily on paleontological studies alone. They also have no consideration of Noah's flood distribution, which describes it to be just a single wave migration. They tell us the first migration wave occurred probably 300,000 years ago and led to the origin of the alpine ibex from the Parisian ibex. The second migration probably took place around 80,000 years ago, leading to the origin of the Spanish ibex. So with their scenario, no close relationship would be expected between these European species, right? However, current mtDNA data indicated that a much smaller genetic distance between these 2 Eurasian species than even between other existing Capra species. Thus, another scenario had to be proposed. After looking at the evidence, they had no other way of getting around the only possible conclusion, whereby the only one wave migration was possible. It came from Europe, following by a speciation process that originated the 2 current species. A paleontological find in Germany gave a detailed description of a Capra specimen which included the characteristics of both modern taxa alive today, supporting the single wave hypothesis and aligning with Noah's flood animal dispersion predictions. Goats are among the earliest animals domesticated by humans, and most recent genetic analysis confirms the archaeological evidence that both the wild Persian ibex of the Zagros Mountains is the original ancestor of all domestic goats today. With even more studies, we have tracked sheep of the wild mouflon, and these mouflon have been traced to the Persian ibex based on the new genetic evidence. The proof comes from the domestication of a bee subgroup in China supported by genetic data which comes from the wild ancestor of the domestic goat, i.e. the Benzora Persian ibex. This is just more conclusive evidence that we can easily track lineage back to a single root ancestor. Since people still do not consider these two related, because one is a sheep and the other is a goat, then I really must give substantial evidence to prove their relation. First, consider sheep are like dogs are to wolves. All dogs descended from wolves, just like all sheep descended from goats. Now, for more proof the mouflon and the ibex are related, the genomes of the wild mouflon and the Benzoran Persian ibex in the Sheep and Goat Domestication Center compared their genomes with that of domestics from local, traditional, and improved breeds. The goat was domesticated from the Benzoran ibex in the Fertile Crescent. This was discovered in 1996. This origin was confirmed by genetic studies based on mitochondrial and nuclear DNA. Also, more proof comes from the documentation allowing scientists to test specific skeletal remains. For those that think sheep and goat are not related, consider this. They can also breed with one another. Their offspring are called geep. Evolutionary scientists ignore genetic data over their fossil record. This is stupidity to say the least. Genetic data in 2005 from mitochondrial diversity data and phylogeographic structure of Chinese domestic goats proved that the Persian ibex was the basal parent ancestor of all modern-day sheep and goats. Also, of the 20 genomic region candidates common in sheep and goat, 14 selection signatures were congruent in both species. Even 4 genes showed pleiotrophic effects and have been related to phenotypic effects between the two. However, since they directly state that they use chromosome B because it evolves less rapidly, and they mix it with the fossil record to adjust and calibrate a timeline, this is all circular assumption based around an old timeline and evolution being true in the first place. It's why they will always fail at obtaining accurate results because they will never consider an alternative. While trying to figure out where the domestication center of sheep was, they began studying the haplotypes. In doing so, they confirmed that the mouflon sheep was the head of all the sheep family, which itself descended from the Benzora Persian ibex of the Fertile Crescent. They tell us today that the Eotragus is the parent basal common ancestor of all creperids. This is all they have for evidence for that, by the way. Like I said, it's all storytelling, no DNA evidence, nothing. They go by a small handful of fossils. That is it. Evolution tells us that modern-day goats, sheep, deer, ibex, antelope, addax, gazelle, water buffalo, cows, and zebu have diverged from the rest of the Bovidea in the early Miocene era, diverging from the Sivirids and the Giraffids from Eotragus. As you can see, they link the tree branches where there was no physical connection. This is how they build their tree of lies. Next, they tell us that the ancestors of the entire genus Capra, aka goat, were goral-like animals, which arose first probably during the Miocene or early Pliocene era. Evidence for this is still lacking. It is just assumed from a single set of fossils which they believe are gorals in appearance. But when genetics tells you one thing and fossils assume something else, you have to go where the biology evidence goes. You may be asking, so what other evidence do they have that these caprid cattle-like ancestors evolved into goats and sheep? Well, taxonomy, that's all. Just classification, not biology, not genetics, or anything of value. I can understand that cladistics is a tool used to make a hierarchy tree of life and is the last line of defense used for lack of actual evidence and the only rescuing device used in their arguments when they have lost, but just repeating hierarchical classification names doesn't actually provide evidence or proof in any way, shape, or form, or that one organism is the actual ancestor of another. It doesn't demonstrate evolution. It demonstrates whatever story you want it to be, or what anyone else wants it to be. There is no reason to assume that nodes of a cladiogram represent punitive ancestors, and without actual evidence besides observational homology of fossils, they have no true evidence, only assumption. And that is what fossils are, pure assumption. It is storytelling at best. It's just like saying those species had something physiologically in common with another, not that one gave rise to another. That is all pure conjecture, based on nothing. The evolutionist thinks it's obvious that if a very small group migrates away from a larger group, that the new group will obviously have less genetic diversity than the larger group that came before it. Yet, the evidence shows otherwise on every study that's been done. Let's look at a couple for just a minute. Let's look at the actual, observable, testable genetic diversity, shall we? Here are some reasons to believe that the simple models are actually not accurate at all. In 1957, a single pair of mouflon sheep were left on one of the Phylogeny Islands near the Atlantic Circle. In the beginning of the 1970s, the number had grown to 100 individuals and peaked at 700 sheep in 1977. Given that the population began with only 2 individuals, the flounder effect, the researchers expected low genetic diversity, measured by heterozygosity. In 2007, the genetic diversity of the ancestors of those sheep was tested and found to be at least 4 times greater than the simple mathematical models predicted. In other words, the models underestimated the genetic diversity of the actual population because they had based all of their models of a preconceived notion that evolution was true. Take away the assumption and you were left with the facts. Other tests of these simplified mathematical models have failed as well. A small population of white-tailed deer introduced into Finland were tested for their genetic diversity in 2012 and again found to have much more diversity than expected based on their simple models. 3 more studies indicating that when direct genetic diversity was measured for animal populations on the verge of extinction, it was much greater than expected, based on predictions derived from the mathematical models. The study involving sheep, bacteria, horses, and gray whales, in which the initial populations were all known, researchers measured the genetic diversity many generations after the initial populations were established. The genetic diversity was known down the line was much greater than ever expected, again, based on models relating genetic diversity and population size. In other words, their assumption method failed validation in each of these cases where all known origin of 2 individuals were conclusively known. If these same models were used to estimate the effective sizes of the ancestral population from the measured genetic diversity at any point in time, they would have overestimated the original population sizes as much larger than 2 individuals. All models underestimated the genetic diversity of the actual population. So if observable data presents one thing and then scientists tell you another, why believe it? Oh, that's right, never mind, don't question anything. Their dating methods of diversity are all wrong because it's based on pure assumption. It presumes long-scale evolution to be true, so they just add it to all calculations. It's untrustworthy, and I prove it because I use their own words. In 406 unrelated animals from 48 breeds of local varieties, the sequence segment spans 721 in relation to the full sheep mitochondrial sequence, even though Sanderson in 2003 used to estimate the time of the most recent common ancestor for each distinct domestic sheep lineage. They still came to the conclusion that mutation rates seem much faster and higher in the recent rather than the ancient phylogenetic history. Ho and Larson 2006. Yeah, no kidding. So they made up a presumed guess using an evolutionary assumption, and it was not accurate. What a shock. But don't worry, the evidence was presented as old anyway. If you take into consideration this and the fact that all evolutionary predictions in the past put against real-world results prove them to be a failure, it is safe to presume that they are completely off on their assumptions of this evolutionary history as well. We all know that they use biased trio dating methods, which assume long-scale time evolution to be true. Now, how about even more direct observational testing methods? You are about to see how biased and indoctrinated they truly are. Look at this. All the represented molecular dates should be interpreted cautiously. They should not be strongly affected by the recent observational nonlinearity and apparent mutation rates, which suggests that mutation rates seem much higher in the recent rather than their ancient phylogenetic history. So, they got results that didn't line up because they were far too fast. Because they instead just assume an old-scale timeline must be true, they went around the actual observable, testable evidence and made up their own. This is just the kind of science you get with SpongeBob universities worldwide. Next, orphan genes. The working assumption had been that, given common descent, and the fact that most housekeeping genes are shared among living things is highly conserved, including the prior assumption that evolution occurs by extremely small changes. Orphan genes should be rare, if not non-existent. However, as scientists sequenced more genes from different organisms, they are discovering that roughly 10 to 40% of each genome's protein coding sequence is new. That is, unlike any other known protein coding sequence. These are orphan genes, and this was one of the biggest surprises to come out of the whole genome sequencing project. Before I can get into it, remember this quote by Nobel Laurel Francis Jacob. He explained the accepted view of how evolution constructed new genes. He said, "Once life has started, in the form of some primitive self-replicating organism, further evolution had to produce through alterations of already existing compounds. As you can see, new genes must arise from pre-existing genes, leaving the signal of ancestry in their closely related sequences, because the probability of an alternative is basically nothing, zero. That is why the discovery of orphan genes, which now show no homology to other sequences, came as a great surprise. It was assumed that getting new genes was hard, and once a workable solution was found, it would be preserved in the descendants that followed. The bulk of genes would have been invented early in evolution, and thus would be broadly shared. However, orphan genes are without detectable homologies in other lineages. Orphans are a subset of taxonomically restricted genes, which are unique to a specific taxonomic level. To put it another way, orphan genes differ from all other genes in that they are lineage-specific, with no known history of shared duplication and rearrangement outside of their specific species or clade. This is yet another way we are able to identify what a created kind is. A human would share orphan genes with primates if common ancestry were true. The fact we share 0% is utter proof that humans were a separate created kind, and creationism is true, not common descent. The more genomes that are sequenced, the more the proportion of orphan genes should shrink if common ancestry were true, as more and more orphans should be shown to be present in other genomes, but that has not proven to be the case. The mountain of orphan genes is growing with every new clade tested, not shrinking. Similarly, horizontal gene transfer was not borne out. The sister genes of orphans should have been found as sample size increased, reducing the proportion of orphan genes. As for gene loss as an explanation, well, it would have been too massive to be realistic to account for the pattern seen. The fact we find orphan genes can only decay but never give rise is also more evidence for our side as well. No testable, observable studies have ever proven that new orphan genes can arise in a species which already had or lost them. The only place this has happened is in the imagination of textbooks. It is also a fact that they are genes without detectable homologies in other lineages, so a fly orphan gene and a human orphan gene are very unique and identifiable. If common descent were true, we should see traces left behind of the orphan protein three-dimensional structure in other species, especially those closest on a taxonomic level, and that is not what we see. Orphan genes are wonderful for creationists and a nightmare for evolution. David Ussery, a biologist at Oak Ridge National Laboratory in Tennessee, and his collaborators compared 1,000 genomes. They found trying to build relations for phylogenetic trees is a waste of time because genetically there is not enough shared information between all life. They state, and I quote, "There are different ways to have a core set of instructions, but of the 1,000 genomes available, not a single protein is conserved across all genomes." The 2018 Gene Research Study looked through 5 million genetic imprints on 100,000 animal species throughout the world to find one of the most surprising discoveries about evolution to date, that they have all been wrong. Evolution predicted that everything evolved up from a single cell, yet this new research looked at a selected portion of DNA from 100,000 different species by hundreds of different researchers from around the world. They found a telltale sign showing that all animals emerged at about the same time. Not only that, the study also found, rather unexpectedly for evolutionists, that species have clear genetic boundaries and nothing much in between. They go on to say in the study that humans and animals arose at the same time and there are genetic boundaries. Thatcher goes on to say, if individuals are stars, then species are galaxies. They are compact clusters in the vastness of empty space. The absence of in-between species is something that also perplexed Darwin, he said. This massive genetic study, which reveals that all life on Earth, including animals, birds, insects, and humans, that all appeared at the same time, should be cause for concern for the evolutionist. Their rationalization and excuses will come soon, but even the researcher himself stated, "The conclusion is very surprising, and I fought against it as hard as I could." How does one explain that fact that 90% of all animal life, genetically speaking, is roughly the same age? Their answer? You guessed it. There must have been an environmental trauma causing a bottleneck that caused all living species to reduce to just a few living number of individual males and females to repopulate. Pfft, ridiculous. So what they're trying to tell you is that this so-called bottleneck, for it to occur, had to reduce all 100,000 different species that they tested to a single maternal lineage to replace all the others to carry the entirety of the linked genome along, thereby resetting mitochondrial variation to 0. Does that sound logical at all? Even in their own peer-reviewed paper, they state that it's not logical. They say, based on contemporary mitochondrial sequence data alone, it is impossible to distinguish an organismal bottleneck from mitochondrial and Y chromosome specific lineage shortening, since both mechanisms make the same prediction of a uniform mitochondrial sequence in the past. So they know and say that they cannot know this, but they will tell you that it happened anyway and expect you just to believe it because. In conclusion, they don't have evidence for this so-called bottleneck, not even their own fossil record shows any evidence of one, and their long time period is all extrapolated from an assumption that a common ancestor exists. They go on to say, for the planet's 7.6 billion people, 500 million house sparrows, and 100,000 sandpipers, genetic diversity is about the same. They would never submit to the actual evidence of the research, which shows that it's obvious that all life arose at the same time. As Kuhn points out, the unresolved arguments tend toward rhetoric. Like all scientists, they struggle to find an example or excuse to puzzle an explanation together before they commit corporate suicide for discovering evidence contrary to the evolutionary theory. They directly state, this study is one of the clearest, most data-rich, and general facts in all of evolution, that the existent population, no matter what the current size or similarity to fossils of any age, has expanded from mitochondrial uniformity. They even directly state that they use the fossil evidence of mammalian evolution in Africa theory to infer an old age of 100 to 200,000 years, based on nothing but assumption, and that sequence analysis has been interpreted to suggest that the last ice age created widespread conditions for a subsequent expansion. Because no actual evidence was seen for evolution, they just had to presume it happened and conclude this by stating, the vista of evolution is best seen from the passenger seat. Why? Because they have no direct access to the driver's seat to witness linear macroevolution occurring. This study was just a perfect nail in the coffin for evolution. Their rescuing device excuses might be enough for the atheist, but it's not for the open-minded freethinker.